So here's a patch of tissue, right? This is amphibian skin, if I'm not mistaken. So we're looking down on some tissue, right? We're going to zoom in as we go here. What do you see? Bunch of cells, right?
Yeah.
What can you tell me about them? So you should already know what this is, right? What is it? It says in your handouts, I know.
Nucleus is
Anybody know what kind of epithelium that is?
squamous.
It's definitely not regimented, right? It's squamous, right? Squamous. Simple, right? Stratified wouldn't look like that, right? So we're looking down on the top of it, right? Simple squamous epithelium. So let's see what organelles we can see here. So we're going to zoom in pretty far here and take a closer look at it as we go. That's about as close as we're going to be able to get self-focus, right? So what can we see? We can see all kinds of stuff, right? Would you agree with that?
Yeah.
If I stop playing with the focus, maybe? Which one do I want to get in focus, though? That's the question. Let's find a different patch somewhere. Yeah, that's not bad. I like that one. Okay. So, what can you see?
Nuclear in.
All right, so what's the pink circles, right? What is this pink circle here? Nucleus, right? It's a little faint, but do you see the dark purple kind of line that's right there?
Mm-hmm.
That's your nucleolus, right?
Specific.
Can you see anything else in there? Obviously, the nuclear envelope is around the outer edge of that. It's harder to see. We'd have to be at a much higher magnification to be able to see it clearly. What else do you see there?
Those mitochondria.
Specs, ribosomes.
The dots.
What are all the little dots? Is that what you're saying? These things?
Yeah.
Mitochondria.
Mitochondria, yes. What else do you see? Do you see the faint purple like lines that look like they're there and kind of go down here? You see that? No? You might be able to see when you look at it on your own microscope. It's ER, right?
No.
So pretty straightforward. What's the other thing you could see in here that is not listed on your list, but is something that's so obvious that you probably don't even realize you're looking at it, right?
Cell membrane.
Yeah.
Cell membrane, right? Obviously, right?
This is the.
Okay, pretty straightforward, right? Nothing crazy. Next one, we're gonna look at something else. We're gonna look at something called a Nissl body. Who was Nissl and what did he look at? Obviously named after someone, yes? Well, first off, we have an issue, right? I think it's on the microscope. You can see those, yes, good, okay. So we're going to zoom in on that quite a bit. Let's look at that guy there, that one. All right. Looks somewhat like the picture, right? So what are we looking at? This is a Nissl body. Nissl bodies are found where? They're found in the nervous system, right? These are nerve cells. So what is the Nissl bodies? In a nerve cell, you have cytoplasmic extensions, right? You can see one going off there, one going off here, one going off there, one coming off here, right? These go into very long, thin little things, right? Which restricts the amount of cytoplasm that the cell has, or constricts it, if you will. Therefore, all the organelles get packed in there really tight. And that aggregate of organelles gives this really grainy appearance to the cytoplasm. And that's what's called Nissl bodies, is that grainy cytoplasm due to the packed organelles in there. Now the other thing that you can see here, and see if I can show it reasonably well. If you look at this cell, you can actually see a real dark center, right? And then there's a light halo around it. So where's the nucleus in there?
Starting at the white.
The edge of the white halo here is the nucleus, right? This is the nucleolus, right? And the extensions, because we don't know which one they are, by convention we call them all axons. If you don't know if it's an axon or a dendrite, you call it an axon all the time. All right, any questions on that? Pretty straightforward, right? All right, the next one is specifically looking for mitochondria. It's a cell that is known to have large quantities of mitochondria in them. Who knows a cell that would have large quantities of mitochondria in them?
Muscle cells.
Yeah.
Hmm?
Muscle cells.
Yeah, but muscle cells would be hard to see. Anybody know another one? Like where this organ's from? Or where this tissue's from? You know what organ that is?
The lung.
Yeah.
Liver.
And the lung too.
All right, let's take a closer look.
Respiration.
Easy to see, right? Piece of cake, right? Nucleus, cell membrane, mitochondria, all kinds of them, big, right? Can't miss them. Have the pages been telling you what the slide says? No. All right, let's go back to the beginning. I'll real quick tell you what it says, right? So the first slide I looked at for page 1 there, it says squamous epithelium, comma, amphibian. The second slide we looked at, page 2, shockingly said Nissl's bodies. What do you think this third slide that we looked at said? How about mitochondria? It's the name of the slide, right? Mitochondria. They won't always be that easy, though. All right, next one. The slide actually says Golgi apparatus, pancreas section. So obviously we're looking at the pancreas, right? All right. So why the pancreas?
Secretes enzymes.
The pancreas secretes lipase enzymes, right? We just talked about that yesterday. It secretes them into the small intestine. So these cells form these little rings, right? So this is a ring of cells. It's not just one cell, it's a ring of cells. Can we get a good focus there somewhere? There we go, that's not bad. All right, so for example, let's pick a good spot. How about this one? Here's a ring of cells, right? That's not one cell, that's about 7 or 8 cells, right? And they all secrete into this central duct, which then goes to the gallbladder, wrong organ. Goes to the collecting duct, down to the tubule that goes to the small intestine there. So how does this cell secrete into that duct, right? Well, it does it by exocytosis, by making vesicles, and you make vesicles with the Golgi apparatus, right? So where are the Golgi apparati? It's this dark purple mass around the nucleus there. Okay? There are special stains that stain for that. Unfortunately, this one isn't very good. Now, a lot of people would have you look at this. I mean, this isn't horrible. You can look at this one. This is, so what, let me go back. So what happened is they stained it, but they didn't stain it well because you got the purple section and the non-purple section. So on this one, you can technically see it a little bit better, but specifically out here, you can see it quite a bit better. And this looks more like your picture, right? And you can see, for example, here, you can see it well lined out that you can actually see the Golgi apparatus there, right? But it's not uniformly across the whole slide, right? Okay. Pretty straightforward. It's not horrible, it's not great. I don't like this slide very much. All right. Now we're going to go to epithelium, right? A few organelles, we're going to look at some epithelium next. We already talked about the epithelium, it's great. It's easy, piece of cake, right? First one, the slide says squamous epithelium, colon, sec, period, Bowman's capsule. Got all that?
Can you repeat it one more time?
Hmm?
Sec, period?
Section.
Yeah.
Bowman's capsule. B-O-W-M-A-N, Bowman's capsule. So where in the world is Bowman's capsule? You guys need me to read it again? Squamous epithelium, colon, sec, period, Bowman's capsule. So Bowman's capsule occurs where? Kidneys. All right, let's find them. Where's a Bowman's capsule? So here's the kidney section. Where's a Bowman's capsule? Not focused very well. There.
Isn't it around the glomerulus?
The what?
It's around the glomerulus? Or is that
What's a
how that
glomerula?
works? It's the tubules all mixed together.
So what we're looking for is we're looking for these round things, right?
Yeah.
So the round thing, the entire unit is called a Bowman's capsule. Within the Bowman's capsule is a glomerulus, which is this stuff on the inside. The Bowman's capsule itself that we're going to look at is the lining of this capsule, which is right there. And we're going to zoom in on that. So you can see it at the same magnification you pictured that. That's pretty close, right? All right, pretty close to what you're looking at on the picture, right? So the glomerulus is this, the whole thing is Bowman's capsule. This lining here, like this, is your epithelium. This is the squamous, simple squamous epithelium that lines the Bowman's capsule. And so remember what I said about squamous epithelium, if you're looking at it on the side, right? All you see really is the nucleus, that's about all you can see. So this one, this one, this one, that one, that one, that one, so on and so forth. Right? Simple squamous epithelium from the side instead of from the top, right? We already looked at it from the top, right? That was the first slide we looked at. And do I need to go back and look at that first slide again? Do you want to look at the first slide again? I can put it back up there and you can look at it again. It's not like it's anything you haven't seen already.
The next one's that, basically.
The number 2 says simple squamous epithelium, which is
Yeah.
the same thing as the first slide, right?
Yeah.
Nothing different, right? It's this slide. Simple squamous amphibian, right? Piece of cake, right? I'm not gonna spend much time on it unless you really want to. We've already talked about that. All right, next one. Simple cuboidal epithelium. The slide simply says thyroid gland. Thyroid gland is very unique looking. It's got a very particular look to it. It's very easy to spot what's going on. The simple columnar, or sorry, simple cuboidal. So the thing to remember about simple cuboidal epithelium is it is secretory in nature. Secretory in nature. All simple cuboidal cells secrete. That's their job. They're secretory. You won't find one that does not secrete. They all secrete, that's their job. So the question here is, we have these, these structures in the thyroid, these little round circular structures, right? Let me zoom in on one a little bit. We'll pick one that has the pink stuff in the middle. So where's the simple cuboidal cells? Those things that ring these, right? There are some that look much better than this one. I don't know why this slide looks like this, but this isn't great. I've seen better. I don't know if I have one sitting around somewhere I could grab or not, but it's all right. Sometimes it looks a lot better than this. This is not the best one I've ever seen. But that's simple cuboidal. Yeah.
What in your eyes gives it away that it's simple cuboidal and not just another side view of the simple squamous? Like
Well, as I said, some of them are better than others. This one is not great. I have seen others where it's very obvious as simple cuboidal cells. You know, look for a thyroid cell, gland cell slide over there. Each tray should have all the same thing on it, so just leaf through and say, no, there's a stack of them there. See if I can find a better slide. Maybe there might be one on my tray, hold on. No. Liver. Oh, nice liver slide. Reproductive, pancreas, colon, about every other thing. Hair. Find one? Let's try this one. Let's try this one. I'll try a different slide. See if it looks better. Ooh, that's worse. That's really nice. Let's not do that. Ooh, that one looks good. I like that one. Let's try that one. This could work. Okay, how about this? Is that better? Much better, right? No?
Yeah.
I think that's much better. So what's the difference, right? You were asked about what the difference between that and simple squamous from the side, right? So first off, you've got the nucleus, you can see the cytoplasm, right? Nice brick-like cells, right? You can see the cytoplasm, right? You will not be seeing the cytoplasm on a squamous cell from the side, right? This is actually not bad for simple cuboidal epithelium. That's about what it looks like. It doesn't really look square though, does it? Never does. Some of them do. If you look over here, they look more square. But most of them look like this. That's the best I got for you. So, there's pink stuff in here, right? And I said these things secrete. So how does that work in the thyroid? So we assume that this is secreting where?
Into the open.
Pink area.
You assume it's secreting into here. And it's just the opposite of that. It's using this to pull that into the cell to secrete into this region up here. This region, this duct region out here.
So the big circles are like storage areas for the
These are the ducts that carry to the bloodstream. Those are blood vessels, actually.
Those are blood vessels? The big ones?
So
This,
Oh, those
this,
blood
this is the stuff that's the pink stuff's in there is called colloid. Yes, colloid. I think. Why am I doubting what I'm saying? Okay. Enough of that. Let's put this over here. All right, so I'm gonna swap out this slide for that slide.
Notice what it has two terms on here, basement membrane and apical layer.
Right?
I was just wondering which was which.
Remember, the basement membrane is where it attaches to the rest of the tissue, right? Where does it attach to the rest of the tissue?
On the outside.
This
Outside.
outer part.
This is the apical layer, basement membrane on that side, right? All right. We're gonna look at some simple columnar epithelium. The slide literally says epithelium, human simple columnar. All right, what are we looking at? Anybody know what this is? First off, it's upside down. Let's flip it over. All right, what is it? It's intestine. So the epithelium is all this stuff up here. That stuff. Let's zoom in on it. Get real close magnification of it. How high do we want to go? There's all kinds of good stuff to see here on this epithelium. Okay, epithelium. Piece of cake, right? So, maybe I'm too far up for you. Let me go back down. Sorry, I'm spinning around on you. Remember villi, right? Villi. So these are long fingers of tissue, right? So when you section it, sometimes what happens is you only get a piece of it, right? So the villi is going to go up and it's not always a straight, right? It's going to go up and curve and do stuff like that. So when you do a slice of it, if it's curved like that, you're going to get a weird section of it, right? And that's what you're seeing here is that you got this one that's arcing this way and you've cut it like so, right? And you're just seeing a piece of it, right? That make sense? All right. So let's take a closer look at that. Okay. So first off, what do we got? Where's the basement membrane?
The inside.
Right here.
Mm-hmm.
Terminal web, terminal bars, right? All that there. Obviously the nuclei, can't miss them, right? Why does it look like there's more than one per cell?
Because on the next cell underneath it.
Cell behind it, one behind that, one behind that, right? This is 5 micrometers thick. You're going to get probably 3 or 4 cell layers thick there on the slide. So what you're seeing is you're seeing multiple layers of cells there. If we could get it thin enough to get 1 cell layer, you wouldn't see that, but very tough to do. And the companies that make these slides are not going to spend the time it would take to do that, right? All right, simple columnar, standard epithelium, we're going to see it a lot. Next one, I'm assuming is ciliated columnar, right? Slide says CIL period columnar epithelium section. Ciliated columnar epithelium section. That's what it looks like on the slide. Any idea?
Is it kidney? Intestine?
Say that again?
Intestine?
Nope.
Oh, is it the lung?
No.
I don't know.
How about female reproductive system?
Ovary.
Oviduct. Not an ovary, oviduct. The epithelium's that red stuff there on the surface. This red stuff here, see it? That's the epithelium. There you go, epithelium. Base membrane down here, cilia is there, you can see it, right? Nuclei are right here, you can see them in that bar right there going across. What else we got? Goblet cells, what's a goblet cell? What's a goblet cell?
Secretes.
Lipids, right? Why do you want oils in the oviduct? Why do you want oil anywhere in your body?
Lube.
To keep it, yes.
Make it slippery, right? You don't, what travels down the oviduct again? I forget. What are those things called?
Eggs.
Eggs, oh yeah, eggs. You don't want them sticking, right? You want them to continue on down. They don't want it, you don't want to implant them there, bad place, right? What do you call that when they implant there?
Ectopic
Exactly.
pregnancy.
Ectopic. Not good, right? All right. Next one. Stratified squamous keratinized epithelium. The slide title is Meissner's corpuscle in VS skin. Meissner's corpuscle in V.S. skin.
What's VS?
Vertical section.
Oh.
As opposed to lateral section.
First one's keratinized, right? That's keratinized.
Yeah, I think so.
Luke is a silly goober.
So this is basically a piece of thick skin is what we're looking at. Thick skin. thick skin like on your hands or on your feet, right? So where is this stratified squamous keratinized epithelium?
Outer part.
Oh, is this
From here, all the way up there is your epithelial layer. So from here to there is your epithelium. That's stratified squamous keratinized epithelium. At some point this semester, we'll talk about all the different sublayers in there. We're not gonna do it now. Base membrane's here. This is the keratinized layer, obviously, right? Stratified squamous non-keratinized epithelium is next. Where's one of the few places where that occurs? There's not a lot of places that occurs in your body.
so this is all first
The name of the slide might give it away.
This is
Skin.
keratinized and this is
The name of the slide says vagina.
Oh, that checks out.
Next one, right? Okay, number 7 there, right?
third, you said?
China.
Yeah.
It's called vagina.
Name
It's
of
upside
science
down.
is
But
vagina.
Okay.
stratified squamous
You don't write it,
non-keratinized
so-
epithelium, right?
So
Nobody
much.
sees that and knows what that says.
I read everything.
I'm just playing.
You have to
Stratified
write it in print
squamous
so everybody
non-keratinized
sees what it
epithelium.
says. However,
Yeah.
you want that right at the
I
top
want
in
it-
big letters.
Yeah,
Is that it?
exactly.
Okay.
Exactly.
Are we ready?
Yeah.
Sorry.
All right. Stratified squamous non-keratinized epithelium. The base membrane is right about there. This is the apical layer here. Lots of nuclei, easy to see, right? Can't miss them. Easy, right? This is thinner or thicker depending on where you look at it. Doesn't really matter where you look at it. All right. Next one. Stratified cuboidal. Pretty much only occurs in one spot, right?
Is that why it's your favorite gland?
What's that?
Didn't you say sweat glands were your favorite?
So what we're gonna do is we're gonna go back to a slide we already looked at, which is the Meissner's corpuscle slide. So this is the Meissner's corpuscle and VS skin slide, right? And we're gonna look for sweat glands. So what do we look at this one already for?
Keratinized, or, yeah.
Stratified squamous keratinized epithelium, right? Now we're gonna look down here in this part of this, this organ, right? We're gonna look down here. This, and that are all sweat glands. So let's look at this one. We'll zoom in on that. So in a sweat gland, there's 2 parts. There's the glandular part and the duct part, right? And I said they're found where? I didn't tell you where they were found, did I? Is it in the gland part or the duct part? It doesn't say, did it?
Gland.
Where do you see 2 layers of simple cuboidal epithelium? You don't see it here, do you? So it's these, obviously, right? And actually, if you focus real close here, you can see 2 layers. It's not always easy to see, but you can see it if you look close. There are 2 layers. So what do you think? Is that easy or hard to see? Would it be easier if I show you on the other part? What did I just say? Where does it occur?
Sweat.
Is it the duct or the gland? I asked you that
Gland.
question.
You didn't answer.
And it's,
Gland. We got
the
gland.
answer is both.
Oh.
Cheating, right? Let's look at this one. Here's some gland right here, right? Is that easier to see or harder to see? 1, 2, 1, 2, 1, 2, 1, 2, right? Is that easier or harder? Depends on how you look at it, right?
Yeah. It's clearer, but you can't
It's
really
clear,
see it
but a
too.
little harder to recognize, maybe.
Yeah.
But it occurs in both layers, right? Both of them, it doesn't matter.
The duct is the darker one, the lighter one with the gland?
This is secretory, this is duct. This is the part that secretes, this is the duct. All right. Stratified columnar, again, a very rare one. We do have a slide here, it says stratified columnar epithelium human, right? Hold on, I'm looking. Where is it? Not loving any of that. Boo. It is. I don't love it. I know what this is supposed to be showing you, but it's not showing you very well, that's for sure. Yeah, that's about as good as you get. It's not great. All right, this is what it is. It's not wonderful. It looks like a mess, not clean. This stuff here. I don't like it very much. That's what it is. Okay. I'm not loving. I never. As I said, it's very rare. Doesn't occur very often, and when it does, it's not easy to figure out. The likelihood of me asking this on the test is pretty small. So don't stress too much about this slide, okay? Transitionals next. Slides labeled transitional epithelium, TS bladder, right? Transverse section, right? All right, so here we are looking at the bladder. So what did I say about this type of epithelium?
Usually you see it full.
So the idea, you're not gonna see it pulled out, right? It's always gonna be looking like it's got multiple layers. You're always gonna see it in this fashion. So the base membrane is down here, this is the apical layer here. Remember what I said, the bottom parts of it, or the parts closest to the underlying skin, are gonna look like they're stretched. The surface areas are gonna be rounded, which is just the opposite of stratified squamous, right? And it's very obvious to see, and the other thing is that the number of layers is not really large, right? There's 5, 6, 7, maybe 10 layers most. Unlike stratified squamous, where you're gonna have hundreds, if not more. All right.
See, this is.
Pseudostratified.
Is that
What?
Space
One of the places I said that you're gonna see pseudostratified is in the trachea.
This is
I
basically.
bet you can't guess the name of the slide that we're gonna look at.
He's pointing to.
Trachea, CS.
No, I don't think that's basically.
Oh yeah, no, that's transitional epithelium.
So we're looking at trachea.
Or cell surface.
This is basically.
Yeah, you're right.
So this is the lowest power. It's kind of an
This is cell
overview of the
surface.
slide, right?
Huh?
This surface cell. Yes, a surface cell. Yeah.
What are you looking at? What do you see? What do you see that's weird? Anything? Or it all pretty simple? Pretty straightforward, everybody see everything, they know everything? That's the whole section, what do you got? So where's the epithelium?
To the left.
Your left,
Top left corner of the.
that side.
Yes.
So he says the epithelium is here, right? No, here,
More.
more. This?
More.
Right there.
Yes.
What's this? Say that again. Cartilage. What kind? Island cartilage. Very good. Why do you have cartilage in the trachea?
Structure. Rigidity.
Keep it open, right? If you didn't have cartilage there, what would happen when you exhaled?
It would.
Collapse.
It would. And then the mucus would make it stick, and then getting air in would be very tough, right? To help with breathing, it's a shocking thing. All right, let's look at the epithelium. Pseudostratified, ciliated, right? No, not there. Hold on. As good as we get. All right. Pseudostratified, ciliated. Base membranes here. The cilia are here. Those little, they're not very long, but they're right there. And the nuclei are all over the place. They're down low, they're up high, they're all over the place, right? You can see here's a nucleus, there's a nucleus, there's a nucleus, there's a nucleus, there's one down there. They're up, down, they're in the middle, all over the place, right?
So,
Pretty easy, right?
how would I tell this apart from regular stratified?
Regular what?
Stratified columni. Or, no, simple columni.
Simple columnar. The variation of nucleus and the cilia are the two keys that you want to look at, right?
So cilia will always be on
Remember, the simple columnar has the terminal bars. It's got the nuclei that are pretty regimented right where they are, right? It's much more uniform looking.
Okay.
This is kind of random. Looks like a mess, right? It's all over the place. Plus, it's got cilia, so you don't really see terminal bars, right?
Okay.
Does that make sense? All right. A lot of talking going on. I take it there's questions because you people are communicating to one another and not me. All right, next one, seriocilia, right?
Yeah.
Why do I have another one of those? Got another trachea slide, different trachea slide. This one is called pseudostratified ciliated columnar epithelium TS trachea, right? Want to look at another trachea slide, see if it looks any better? Oh, this is prettier by far. This is a different section, right? What was the other section? The other one was CS, cross section, right? This is TS, transverse section. So what do you see first off? What do you see about the cartilage? Instead of that ring of cartilage that you saw around it on the last one, what are you seeing?
Balls. Cartilage.
cross sections of it, right? And then let's look at the epithelium. Look at that. I think the epithelium on this one looks much better than the other one. Plus, it's a trichrome stain too, which helps a lot. 3 colors instead of 2. There's your epithelium. How's that look compared to the other one?
Way better.
Where's the cilia?
Are those goblet cells or?
Yep, those are goblet cells.
Okay.
There's a good section of cilia there. You see them
Right.
pretty easily, right? Zoom in a little bit more. I'll show you a little bit higher magnification.
So the blue parts are goblet cells?
Yep, the blue part of goblet cells. Cilia is real easy to see right in here. Look at those nuclei, everywhere from down here on the bottom to up there, right? All over the place. What's this? No idea. Some stringy looking stuff, right? We haven't gotten there yet. I just thought I'd ask. No guesses?
Is it
Smooth muscle.
smooth muscle?
All right. Next one, stereocilia. And I don't see the slide I want for that. Is there an epididymis slide over there? Oop, found one, got one. So this is for the pseudostratified columnar epithelium with stereocilia, number 12, right? The slide literally says epididymis, parentheses, human, comma, section. So the first thing we have to do is orient the slide a little bit. So there's 2 types of tissue in here that you can see. There's this one and this one. You gotta tell what it is. Where is epididymis here? Is this epididymis or is this epididymis? What? This?
I don't know.
The other
I would
one?
agree. That's epididymis. What's the other stuff?
Oh,
Where's the epididymis?
Tubules?
Seminiferous tubules. Testes. Right? So this is testes. This is seminiferous, or epididymis, I'm sorry. Seminiferous tubules are testes. So we're looking at pseudostratified epithelium with stereocilia. Remember, stereocilia are long cilia, right? So we're going to zoom in on this epithelium, which again is this stuff here, this stuff, right? Stereocilia are somewhat hard to see. See if I can find a spot where you can actually see them. Hang on, I'm going to be roaming a little bit so I find what I'm looking for.
Are those free floating dots anything?
Yeah, we'll talk about them in a second. There you can see it a little bit. Let's see, there's a couple of them there. They're very wispy and very hard to see, but you can see, and commonly what happens, they get ripped off when you section to pieces. So they're very hard to catch, but every once in a while you'll find one. There's one there, there's one there, another one over here. Those are stereocilia. They're very long, very thin. You don't see them a whole lot, but I will show them on the slide when you see them. The question I had was, what are these weird dots in the middle, right? This stuff down here, right? This stuff. You might know what that is?
The sperm.
It's called free spermatozoa, right?
Would you go back up to the, like, where there's like a little yellow thing? What was that?
Will I go up to the little what thing?
If you, like, scroll down, there was like a little yellow,
A little
like,
yellow
cells.
thing.
Hang on, right there. On the left. My left.
That?
Yep. What is that?
Artifact,
Just artifact.
It's just
Okay.
garbage,
Cool.
junk.
I didn't
Who knows what that is? Don't know, don't care,
Cool.
right?
Yep.
You see them every once in a while. You get some kind of weird thing in the process of making the slime, right? All right, we'll talk more about epididymis when it comes to that part of the class. Where are we? Exocrine glands, we're gonna look at glands. Unbranched, simple tubular exocrine glands found in intestinal glands, the intestines, blah, 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 blah. Really? So, really? So this slide is small intestine duodenum, duodenum, whatever way you want to call it. The technical term is duodenum, but you can call it duodenum if you want. So where are the glands I'm talking about here in that picture? There's actually two sets of glands here, right?
The clear circles, semi-clear.
So we have these are glands here, and then these are glands as well, right? Which ones do you think are simple?
The bottom ones.
Probably these, right? Those are not simple, right? Those are compound, right? Easily seen to be compound, right?
So those are branched.
What number am I on?
These are branched.
Two.
Number 2, branch tubular in the stomach. We're gonna look at the stomach now. This slide's labeled fundic stomach. Fundic. It's also a it's a TS, by the way. Not that that matters, but this is a trichrome stain? Yeah, trichrome stain. Okay. So what we're looking at is where in all the glands. Where are all the glands in the stomach? The glands are this is all the glands here. All that stuff, right? Those are all your glands there. From here down to there. And those are the branched simple tubular exocrine glands. It's also got one of my favorite cells in it, right? That I mentioned already in class, and I will mention them many more times. What did I say they were?
Sweat glands?
Parietal cells.
No.
What did I say about them? I said they're bright red. Which ones? Anybody see them? See bright red
Yes.
cells? A couple. They're all over the place, right? Bright red cells, parietal cells. And what do they secrete? What did I say they secrete?
I don't know.
What? How does your stomach break down
It-
food?
Acid.
Acid. What acid do you think they secrete? Hydrochloric
Chloric
acid.
It keeps like the lines on our diagrams. Which one is where are the
circles?
The secretary units in this? How do I recognize that?
Each one of these cells is the secretory unit. Every little circle, right? See the little gaps in them like this?
Yeah.
That's a duct, right? And the duct simply goes up to here, right? It doesn't go very far.
So is
For
it,
example, there's a big one, right? You can't miss that one.
is it a
Everybody see the duct? This one?
if it's a branched, is the secretory unit like all of the individual cells put
Every
together?
single cell is a secretory unit.
Oh, okay.
And they secrete into that tube.
Okay.
Think, kind of like grapes, right? Each grape would be a secretory unit going into the stalk, right?
Yeah. Okay. Makes sense.
Coiled tubular sweat glands. We're gonna go back to that slide. What slide was it? Meissner's corpuscle. Remember that one? How many times have we gone back to it already?
Twice.
We're gonna use it a lot in this class. It's one of the best slides I have. It shows probably 20 different things in it. There are certain slides that show a lot of different things. We're back to sweat glands again. Wait a minute. We already talked about this. Do we need to talk about it again? You know the secretory and the excretory unit, right? We don't need to talk about it again, do we? Or do we?
For fun.
For fun?
The denser
That doesn't
one
sound
is,
like-
the dense one is.
How many
the
times have I
duct.
done this?
Dense is the duct.
Is it fun for me still?
Yes.
Yeah, I feel like
If it wasn't fun, I wouldn't do it, right? All right. So
it
there's the sweat gland.
is. Okay.
This one's. the
So you've
duct.
got this part
Duct.
and this part are the same thing. These two are the same thing, which are the secretory part, right?
Mhm.
These are the ducts, right? So it's a coiled tube, goes all over the place, right? So when you cut it,
Is this
you get little pieces of it everywhere, right?
Yeah.
Yeah, that's
Does that
all
make sense?
secretory.
Notice that some of them look like this, right? Because you got a curve going and you cut through the curve, right? I don't know, you wanna look at another sweat gland? How about this one? Is that better? What's this? Duct?
Ducts.
Secretory unit.
That's the secretory, no, not secretory, this is duct, right?
Duct is denser. Right?
What's that?
Fat.
God,
Adipose. Yep.
Adipose. Yeah, that does look like adipose, doesn't it?
All right. Good enough? All right. Put that back where it belongs. 4. Compound secreting gland. Hey, look, mammary glands.
There's no lines going on.
What do you think? What do you think the slide says?
Mammary gland.
Mammary gland. VS lactating. Why do we have a lactating mammary gland?
So it's
So it's
working.
So you can see what's going on. So this is a secreting mammary gland, big gland, right? That's all the mammary gland is a big gland. It's actually technically a modified sweat gland. I don't know if you know that or not.
Oh shoot, that's so true.
So what's all the pink stuff?
Similar to what we saw
Duct.
on the earlier slide.
Duct.
Product, right?
Okay.
What do you call the product of mammary gland again?
Milk.
Ah, milk. How about that? Milk, right?
Oh,
Oh.
pink stuff is milk.
That's not what I thought you were
Oh.
about. Okay.
There's tons of it, right? All this pink stuff.
Because it's full of fat.
So where's the secretory units? It's all these cells that line these, right? The ducts, these are the ducts here.
Where are the ducts? Sorry.
Ducts.
That's what
Here, this is a little bit better. You can see the ducts a little bit better here. Everybody see the ducts there? Pretty strict. I mean, it barely shows you kind of where they are.
Kind of backwards.
What would it be if they weren't lactating?
just not have
Smaller,
anything.
much more compressed, right? Wouldn't be any pink stuff, right? All right. Compound acinar exocrine gland. This slide is human scalp vs hair. It's long, right?
Humans.
You guys caught up? Do I need to stop? Go back? What?
We were just confused on that one way back.
What's that? You weren't done with that one.
Yeah.
Is it
so-
too simple?
No.
It's not hard. That is simple. Lots of gland, very little duct, and that's about all there is, right? There's nothing else really. A little bit of blood vessel, that's about it, right? There's not much else.
Excretory.
The what?
The large ones for the excretory.
The large one.
Yes.
That's what she's talking about. Hold on. Now we gotta go back. It's all her fault, people.
Thank you.
So what are you asking? Be clear if you can.
There was a bigger one that, I think.
There was a bigger one.
I don't know, the
Wait a
larger
minute.
ones are the excretory ducts.
Don't try to lead me into this place.
The large blue
I don't need to
tunnel
make jokes like that.
is excretory.
What are you asking?
Which one was the excretory duct? That's what we were,
All right, so this all is gland here. All this is gland.
okay.
All of that, right? This is your duct here, right? This is your duct here. That's it. I mean, the vast majority of this tissue is gland and duct, right? There's very little else. The only other thing you see is a few blood vessels.
Where are the blood vessels?
Where's a blood vessel? So for example, let's see, blood vessels. Not that you would tell that it's a blood vessel very easily, but here's one here.
Oh. Interesting.
See the little section of blood in it right there? There's one here,
Right there.
there's one there, there's one there. How long have I been teaching this class? Histo, first time I taught histo was 89. 89,
Human is
35 years.
whatever that is, I don't know, whatever. Forever ago. Nobody here is 35, right? Does that answer all the questions, or do I need to do more? I could zoom in on it and show you stuff, but it's pretty simple. All right. Are we ready? Human scalp vs. hair insertion sebaceous gland. Long title, right? Human scalp vs. hair insertion sebaceous gland. Long title. I should make a list of these. No, that'd be like work or something. I can't do that. That'd be like teaching. I can't do that either. All right, so we're looking at skin. We're looking at where the hair is, right? Like your scalp, right? So where's the hair? I don't know. What's all this weird stuff?
Is that her?
Where's the epithelium?
Right there. With that clear circle. Oh, it's on the outside?
It's up there. That's epithelium, right? What kind?
Stratified squamous.
Columbus.
Keep going.
Keratinized.
Keratinized. Very good. Doesn't look like thick skin though, does it? That's thin skin, right? Your scalp is thin skin.
That's what you have, Maverick.
What?
So where's the hair follicle?
Thick skin?
Thin
Do you
skin.
see a hair follicle?
Oh, thin skin. Take
What's
as a
that?
Compliment.
What's this?
Is that
Hair.
Hair follicle. That's a hair. What's this?
Sweat gland?
Sweat gland. Sweat on your scalp, right? What am I looking for? Oh, sebaceous glands. Somebody's already used that term. They already know what a sebaceous gland is. Right? What's the space? What time is it? How's my time doing?
Great.
What time is it?
11:20.
Secretes
We got
sebum.
like a half an hour left. I got tons of time. Can talk for another half an hour.
This was this one.
So what am I looking for?
Sebaceous
Sebaceous
gland.
gland.
I'm gonna talk about something else. It's not gonna be on this test, but it will be on a later test that we're gonna talk about. One of the other structures that's in here that you're gonna see is, and let me see if I can find a good one. That one's okay, it's not great. That's a good one, but you can't see what it's attaching to. Oh crud. There's no really good one here in this picture. So what do you notice about these hair follicles?
They don't have hair in them.
They're not at a right angle to the surface, right? Your hair doesn't stand straight up, right?
Mm-hmm.
It kind of lays down like this, right? Like that. And sometimes it goes like this, and sometimes it goes like that, and that's when you have trouble with your hair, right? If it's not all going in the same direction, right? But what did you know your hair can move?
The piliary. Yeah.
You got little muscles there that can make it stand up, right? They're called erector pili muscle. Smooth muscle bundles. Let's see, where's one? I see one there. Hold on.
Is this the
There's one
thing
there.
that you're saying is gonna be on a later test?
It'll be on a later test, I guarantee it, but it's not on this one. So that makes your hair stand up. Why do you want your hair to stand up?
To get rid of heat, or to contain heat, reserve it.
On us, it doesn't do a whole lot, right? It doesn't help us very much. And in fact, we have parts of our body where if you still have hair follicles, but not very many, right? Unlike most other mammals, right? We have hair follicles, but not very many. And so if you have those muscles contract on most of your body, like if you're cold, what happens when you get cold?
Oh, goosebumps.
Goosebumps. Goosebumps are those muscles contracting. It's like flexing your skin muscles. It's very weird, right? The Danish people have a different name for it. They call it myrintiges. But anyway, that's what that is. Goosebumps are your little erector pili muscles bundling up and trying to cause the hair to go up. So what if you had fur? What if you're like a dog, right? If a dog gets cold and they contract, what happens? All that hair stands up. What's it do? It makes the distance between the skin and the air further apart and helps keep them warm, right? There's another aspect of this other than heat control, which is what? Protection, right? If they're going to get into a fight, they do the same thing. The hair goes up because it makes a longer distance for that enemy to get a weapon, whatever that weapon is, teeth, whatever, through the hair to get to the soft tissue underneath. So erector pili muscles, we'll beat that in the ground. So let's talk about the other thing, the gland thing, compound acinar gland, right? Here we're talking about sebaceous glands. There's one there in the field of view. Where is it?
At the, yeah.
Sebaceous glands, right? Let me go see if I can find some more. There's plenty of them in here. How about that one? That's not bad. I like that one. Here, 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 there. Right. Lots of sebaceous glands. What kind of secretion is it? It secretes something called sebum.
Oil.
It's oily. So this is so what happens is as your hair goes up and down, right, with the erector pili muscles, that contracts against that sebaceous gland and makes it secrete. So the more you get cold and warm, the more that oil gets secreted out. Why do you want to have oil on your hair if it's cold outside?
To retain heat.
What does oil do? So you got your hair follicles, right? And they happen to brush against each other and you got oil on them, what happens? They stick. They make a nice solid mass, right? Help with that temperature control, right? If you don't wash your hair for a long enough period, your hair becomes a big clump from that oil, right? So actually, if you don't do anything to your hair and you don't shower for a very long time, you end up with this big, like a helmet of hair.
Let's go.
Right? And actually, that's a pretty solid defense, right? both for warmth and for that. So why don't we do that today? Why don't we do that in our culture today?
It's not pretty.
It's not.
Because it's stinky, right? Would you agree with that? It's gross, it's stinky, right? You're not clean if you don't take a shower every, 6 months or whatever. You know that, yeah, I'm not gonna go into,
Not every day.
take anthropology and see what they say about it, right? There's some really interesting customs we do that are associated with how often we bathe, or used to bathe. Like bouquets at weddings, right? Why you have weddings in the spring, all that stuff, right? You understand what I'm talking about?
Yeah.
Used to be that weddings were always in the spring, way back when. They were always in the spring. Why? Because that was one of the 2 times in the year you took a shower or a bath.
I thought it was just because the weather was pretty. I don't know.
The bouquet was to, cut down on the aroma. So what do we do with our hair now that we don't let that oil in there, right? What do you do?
Shampoo and conditioner.
So you shampoo in to get the oil out, right? Then when you put conditioner to put the oil back in.
It's a crazy cycle.
So you don't like your own oil, you're gonna put other oil in there.
Yes.
Right? Does that make any sense? So you use one product to take it out, another to put a different one in.
If you want to get picky.
And then you complain about split ends.
Ends to ends, but,
pH balance as well.
What else do you do? Same thing when you shower, right? You wash off all the oil off your skin, right? With soap.
Put lotion.
Then what do you do? You put lotion on. Okay, that's brilliant. All right. We're off in the weeds now, that's for sure. All right. Okay. Where am I? Oh yeah, salivary gland. Okay. Last one, salivary gland. Guess what the slide says? How about tongue?
Oh, touche. Makes
Tongue,
sense.
VF, taste buds. Tongue, VS, taste buds. These are the glands down here. Can't miss them. Right? Big glands. There's the glands, huge glands, right? Can't miss them. What's that other stuff? What's this stuff?
Muscle.
Muscle fiber.
Muscle.
What kind of muscle?
Skeletal.
Skeletal muscle. Your tongue is under voluntary control, right? You can move it. Hopefully. Then we can go into genetics of whether you can do certain other things with your tongue, right? How many people are rollers here in the classroom? You know what I'm talking about.
Yes.
Everybody's had that class, right? If you're a roller, stick out your tongue. Roll it. How about you non-rollers? You don't want to be called out for being a non-roller? You think that's an inferior situation?
No, I do not.
Well, there's another genetic one that's always good in this same venue, and we'll get away from the tongue a little bit. It has to do with winking, right? Some people can't do that. Anybody here cannot wink?
I can only wink on one side.
There are some people can't wink at all, just can't do it. Both eyes will do it, right? They can't separate the eye.
I wanted to be able to.
All right, so let's go to the surface, talk about taste buds a minute, and then I'll call this here. So taste buds are up here on the surface of your tongue, right? And everybody I'm sure has heard the thing about certain regions of your tongue taste different things, right? Which is relatively true. So taste buds are these things, right?
Thank you.
But what I want to talk about is I want to talk about your nose, not your tongue. So everybody knows what tasting is. You put chemicals on it, you taste it, right?
Right.
What about if you smell?
Same thing.
So your glands that smell in your nose are modified taste glands. So when you smell something, you're tasting the air. Remember that the next time you smell something really bad, that you just tasted it, right?
So does that imply that taste came before smell?
That's what it implies.
Cool.
Evolutionarily speaking, yes. So, the other thing weird about us as a people is that we are very courteous to our friends, right?
Occasionally.
Anybody know what I'm talking about? You ever heard of a courtesy sniff?
A courtesy sniff?
Courtesy sniff.
I have not.
So,
You don't have good friends.
here's what you're gonna do, right? So the next time you're with some of your friends, not from this class, right? When you're with some of your friends, you walk into a room, the first thing you gotta do is stop and go, oh, something stinks in here. I don't like it, right? What are they gonna do? Everybody who's with you is gonna stop and do what? Courtesy sniff, right? Guaranteed.
Okay.
Do it and watch. Human behavior. There's not one of them will not do that. They will all do it, every single one. Ways to mess with your friends, right? No? He's gonna go do it with his brother, what?
I will.
All right, so there were a lot of pages. This was a fairly large lab, unfortunately. Some of them will not be this big. There are a few of them that are like 2 or 3 pages. Some will be very short. The problem is that on these, we had 1 or 2 things per slide. When we get later on, there'll be 10, 20 things per slide that we have to do as we get further along. What's up?
I was just wondering what goes in the organelles, epithelium, and glands lab submission on Blackboard.
Well, that was if you wanted to submit it electronically.
Just like what we wrote here.
Yeah,
Oh,
you're supposed to label everything.
Can we go? Yeah.
Can we go over to what?
The last page in the lab.
The last page of the lab, which is what?
The taste
The
test.
tongue?
SNR one.
This one?
Yeah. Can you point out the ducts?
Can I point out the what?
The ducts and stuff
Okay.
and the glands.
The ducts and the glands in the salivary glands in your tongue.
Yes.
Compound tubuloacinar exocrine gland, that one? Number 6?
Yes.
Hmm.
But it has to be 2 different glands, the tubular and the one.
Yeah, what about them?
We usually know which one it is.
Okay. So what's the terminology? Tubular, right? Remember tubular, right? What did I say about tubular?
Long, like a tube.
So what is this called? Tubuloacinar, right? What did I say they were? You gotta,
It's got
remember
a
the
ball
picture
at
in
the
the
bottom.
lecture?
It's got a ball at the bottom.
And a
of the
ball
tube.
at the bottom, and then the
Oh.
duct going up, right?
I see.
Right? And the cells down here, like that, right? So, what's this one?
Is
That's the ball at the end, right?
ball?
That makes sense? What do you think this one is?
So that's asinine.
So here's the ball at the end. What's that?
Hmm.
Tube, right? That make sense? Here's another one, right? Ball at the end, tube, right? Are you confused? Look at this shape. Where do you see this shape? You see it here, right? If you turn that like this, right? Here's the ball at the end, there's the tube, right? Make sense? No? Are you confused still? How about that one up there? Hold on, let me pull that down to the middle of you.
Ball on the ends on the right.
How about this guy? Ball on the end, tube, right?
Cool.
I'm guessing. Big guess.
I think I found one that will work.
Is that better? No? I don't know if I can find one better than that, maybe.
And the ducts are just the parts in between all of them.
Well, here's a good example. Duct, right? Duct. Now I'm really gonna mess with him. You ready for the fun here? What's this? No idea. What's this here?
Epithelius.
Is it smooth muscle?
Squamous.
Nope. What'd you say?
Squamous cells.
Nope.
Is it? I don't know.
All at the ends that are bigger.
Is it fat?
It's smushed.
Should I go down a power so you can see it better? Let's see. Does that help?
Could you reduce the light a little bit?
I can, but it won't do anything. Notice how it readjusts? That's the problem with auto adjusting televisions, is that I can play with my light all I want, it's gonna go back to what it was. So what's this thing here? Nope.
It's that thing. Never mind.
You got an idea, it's just not coming out.
Yeah.
Nobody.
Is it, I don't know, a receptor or
It's not the.
temperature?
You guys want me to give you, I'll tell you what, no phones, no cheating, right? If you can tell me in the next 2 minutes what it is without me telling you, I'll give you an extra point today.
Is it,
No cheating. So I've had lots of guesses, right? Smooth muscle, no. What else did we say?
Excretory tissue.
Not excretory tissue. Why would you have, no, not excretory.
Not transitional.
Endocrine.
Why would you have excretory tissue in your tongue? So,
Amylase.
you're talking about the excretory duct maybe? No, it's not the duct. Amylase.
Amylase is the enzyme in the mouth that breaks down the starches.
That's what these things are secreting. I can't hear you.
is it,
No, not
is it a
salivary.
nerve?
Ah,
Wait, is it a nerve?
it's a nerve.
Hey! So I get an extra point.
We all get an extra
That's a
point.
nerve
Just
bundle.
kidding.
Nerve plexus, as it's called.
Nope.
Very good.
They're like that, so never guessed it.
How would you know, like, what key features do it?
You know, I've been looking at them for years, and all I can say is I see what they look like. We'll talk about it a little bit more, but it's kind of a funny look to it, right? There's a classic look to a nerve bundle. All right, that's it.
Can we turn this in?
You can turn it in, yeah. Just set it right there on the end if you want to turn them in. If you want to turn them in the old-fashioned way, put them right over there. Oh, the goblets all over.